Hi, have you ever wondered how those companies like Ancestry.com are able to just take a small swab of cheek cells and then from that work out your relationship to lots of other relatives and also people and cultural groups from around the world? Well, the reason that they can do this is that we can extract DNA from small amounts of samples, like just a few cheek cells, and then copy or amplify that to make enough DNA to do some proper analysis. They obviously can't sequence your whole DNA for 50 bucks a kit. So instead what they do is they pick out particular markers that are found throughout the DNA, use those in their analysis. And that's the kind of stuff we're going to be talking about in the next few weeks around biotechnology, starting with this one, PCR, about polymerase chain reaction, then talking about DNA fingerprinting, profiling, sequencing, and all sorts of other molecular techniques. So in terms of the student learning outcomes, what we have here is you need to learn that DNA can be extracted from cells. Now that's pretty straightforward. Whoa, low battery. All right, that can be done through lots of different means, physical and chemical. And then we can use these modern techniques to analyze very small amounts of DNA, particularly by using this technology, PCR, to multiply that segment of DNA that we want to analyze. And that the components of PCR are primers, heat-resistant enzymes, and nucleotides, and we use the process of heating and cooling in order to make that into multiple copies of DNA. So let's just have a bit of a look at how this works. If you're going to analyze some DNA from cheek cells, for example, you need to extract that DNA and copy it before you can analyze it. This copying of DNA is also sometimes called amplifying, and the main technique for that is PCR. So here's some of the processes around how you might go about that. But really the key thing is that PCR is a way of copying DNA, just like DNA is replicated in the nucleus, only in this case, you're doing it in a small test tube, a PCR tube here. Now think about what are the components in the nucleus when that's happening. Obviously you've got the DNA sample or the template DNA that's being copied. You have nucleotides and you have a DNA polymerase enzyme. In this case, we're talking about Taq polymerase, and I'll talk about why it is that later on. But that's a DNA polymerase. Copying this DNA sample using the nucleotides. And if you might recall from DNA replication, go back and have a look, is that there are primers that need to attach to that DNA sample first in order for the DNA polymerase to work and start to elongate that DNA. So those are some of the things that are contained within that tube. Then there's a heating and cooling cycle that's repeated many times in order to copy this. So you start from 1 copy to 2 to 4 to 8 to 16, exponentially increasing the amount of DNA. So what is this cycle and why do we call it heating and cooling? Okay, so the main difference, so although the ingredients are kind of similar to what's in the nucleus, the way in which it happens is quite different, because the key thing here is that rather than it being part of a cell cycle of replicating DNA, PCR does this cycle every few minutes in order to increase the number of copies of DNA very quickly, and it does that through a temperature cycling process. So what we have here is the strand of DNA that you're interested in copying. The red's the primers, the blue's the free nucleotides, and of course you need to separate those strands out before you can do any copying. Now in the cell this happens through helicase, unwinding that DNA, but in a PCR machine we use what's called a thermal cycler that will heat up the DNA, just in the same way that heat will denature proteins and get their folding to come apart. Here heat will get those 2 strands of DNA to come apart. The heat energy is greater than the hydrogen bonds holding them together, and the 2 strands will separate. Then you cool it down to cause annealing. Now annealing is just a technical word that is about when the 2 strands come back together. Now sometimes those 2 strands that came apart would come back together again, but most of the time what happens is these short primers, which are complementary to regions on those strands, will bind there first. So the DNA comes back together but made with a new partner. Then you warm it slightly to, in this case, about 72 degrees, and the DNA polymerase will copy until it reaches the end of that template. And now you've gone from 1 double-stranded to 2 double-stranded copies. So now there's 2 copies, and then you can heat and then cool and then warm again, and end up with 4. And you go through a process each time this gets heated and separated Okay, I'm back after a short technical hitch there. Battery's full again, and yeah, so what we're looking at here is that those 2 copies go to 4, and this continues as we go through those cycles to go through that denaturation, separating the strands, annealing, bringing in together the template and the primer, and then elongation as the polymerase adds free nucleotides to copy that. What you end up with is, despite any length of the original template, it's just the sequence between those 2 primers that gets amplified. So these original strands become obviously proportionally less and less of the sample, and you get mostly what you're copying is just these short segments in exponentially increasing numbers. One thing just to come back to, as I said before, heat-resistant polymerase. So you may be wondering, denaturation here of that DNA, separating those strands, surely that temperature is going to denature the protein as well. And what protein are we talking about? DNA polymerase is a protein, obviously, an enzyme that needs to keep working in order for this polymer to be made. So how is it possible to have a DNA polymerase that can withstand 96 degrees Celsius? The reason is scientists, biochemists scoured extreme environments for enzymes that work at very hot conditions. So this particular polymerase comes from a thermophilic, a heat-loving bacterium that lives near hot springs and can withstand very high temperatures that come from that geophysical energy coming out of the hot springs. And so their sequence of amino acids has evolved in such a way to be such a stable structure that that polymerase works even under those extreme conditions. Just to add a few things then, there's a couple of videos there made by Invitrogen, the company that makes PCR machines. There's a number of different ones that are available on the market, but these guys have made some pretty straightforward, simple videos, and there'll be some more resources there as well.